Step 1. Get a Ziploc bag and a strawberry, and you have to take all of the green pieces off of the strawberry. You put it into the bag, and you take out all your frustrations by mashing. Now that you've mashed all of your strawberries up, we are going to try to break the cell membranes and the nuclear membranes by adding this lysis solution. This lysis solution has some dish detergent as well as some salt, and that will help to isolate the DNA and to break those membranes. Now that you've mixed it again, we have to strain out your solution. So we are going to be using a cup and a coffee filter. Make sure that what you have at the end is nothing but juice. You can't have any pieces of strawberry left. So in your cup, you should have just the strawberry juice and no pieces whatsoever. For the next step, you will need a test tube and some isopropyl alcohol that is pretty cold. So you're going to take your strawberry solution, pour it into your test tube, make sure it only is about 1/3 of the way full, and then you are going to slowly drop in some isopropyl alcohol, making sure to put the tip of the pipette over on the glass so that the DNA immediately starts to spool. All right, and as soon as you start doing this, you should be able to see a line where the DNA starts precipitating out. It will look clear and quite honestly like snot.
Okay, can I go there?
All right, now in between the strawberry and the alcohol, you see a very light pink layer, and that is going to be where your DNA is, and now we can spool it out. All right, so now if you have done it correctly, you should see a line in between the alcohol and the strawberry juice that is a very light pink color, and we should be able to take a bamboo skewer now and spool this into an Eppendorf tube that already has some alcohol in it. So now, if you have followed all of these directions properly, you should have your DNA in your Eppendorf tube. And the reason why we use strawberries is because strawberries are what are called octoploid, which means they have 8 copies of every one of their chromosomes. So where we have 2 copies of chromosome 1, 2 copies of chromosome 2, they have 8. So unless you really didn't follow instructions, you should have gotten some DNA.